we've learned so far has been called C3 photosynthesis. And this is basically just because of what Rubisco does with carbon dioxide. And the basic thing is that Rubisco, as carbon dioxide comes into those leaves through the pores and mostly the bottom of the leaves, the stomata, is Rubisco is going to grab that carbon dioxide itself, and it's going to bring that carbon dioxide that it grabbed from the atmosphere and bring it into the Calvin cycle, where we're going to end up making carbohydrates and ultimately molecules of G3P that will ultimately be turned into things like apples and cellulose and glucose molecules for whatever the plant particularly needs. So in C3 photosynthesis, this is plants where Rubisco is taking carbon dioxide directly and attaching it to the Calvin cycle, where we're doing carbon fixation, reducing it, and then we're getting G3P molecules out that we could build into things like apples, cellulose for cell walls, wood, and leaves, fruits, vegetables, seeds, starches, and these sorts of things. Now, the plants that are going to do this are pretty much most of the plants that we traditionally see around us when we look outside a window, things like most of our trees, our traditional grasses on a lawn, and our shrubs. Now, C3 photosynthesis does have a few drawbacks in certain conditions. And one of those is that these plants are going to sometimes experience some higher temperatures. It's not going to be high enough to denature their enzymes like Rubisco, but it is going to be high enough to cause issues with water loss from the stomata on the bottom of those leaves. So when things get hot, what we see happening is we see water being lost from those stomata, especially underneath the leaves, and that water is leaving because of transpiration. And that water is being lost and going out into the atmosphere. And that obviously reduces the amount of electrons that the Z scheme has to operate. So there's less electrons going to the Calvin cycle, and that's slowing the formation of sugars down in these plants. So that's one issue with warmer temperatures in C3 photosynthesis is there's a water loss problem through transpiration. Now, the other issue when temperatures get pretty warm is that Rubisco here is going to get a little bit confused. Instead of grabbing something like carbon dioxide, Rubisco's going to end up grabbing oxygen. And if it's grabbing oxygen, it won't be able to take any CO2 into the Calvin cycle, and we won't be able to get any G3P formed, and we won't be able to form any glucose. Instead, what happens is that carbon dioxide stays into the atmosphere. Rubisco is grabbing that oxygen molecule, and it's going to take that oxygen molecule and send those oxygen molecules into the Calvin cycle. But what's going to happen is when that oxygen gets in there, there's no new carbons to rebuild RUBP. So what's going to happen is those carbohydrates in the Calvin cycle are now going to produce energy. And that's great, except it's not energy the plant can store. It's only some ATP it's going to produce at that moment, because it's going to take those carbon molecules and send them into photorespiration, which is kind of like a cell respiration pathway. So the solution that some plants have for photorespiration at high temperatures when it's hot outside is they've kind of come up with a different way to get that carbon dioxide molecule and make sure it gets to rubisco and it can enter the Calvin cycle. And this is known as C4 photosynthesis. Now, what's going to happen in C4 photosynthesis is we're going to change who grabs carbon dioxide first out of the atmosphere. In this case, it's this truck that we have here, and it has a name, and its name is PEP carboxylase. And what it's going to do is as carbon dioxide comes into those leaves, it's going to grab that carbon dioxide, and it is very much like a pizza delivery vehicle going to take that carbon dioxide, and it's going to deliver that carbon dioxide directly to rubisco, kind of like a pizza delivery. And it's going to hand deliver that carbon dioxide right to rubisco, and rubisco enters it in the Calvin cycle. We're not bypassing rubisco, we're just changing in C4 photosynthesis how rubisco gets its carbon dioxide. In this case, PEP carboxylase is doing the carbon fixation, and it's taking carbon dioxide out of the atmosphere, handing it to rubisco, and rubisco is entering it into the Calvin cycle, where we can use it to make some carbohydrates and produce G3P molecules. Now, the types of plants that are going to use this are the types of plants we typically kind of see doing very, very well around here in Windsor, Essex during the summer. Things like corn or C4 photosynthesizers. They grow very, very fast over a couple of months. A lot of the weeds we might see in our lawns and our gardens, they're also C4 photosynthesizers. They do great in really, really warm temperatures as the growth of other things starts to slow down. Now, the other situation we have is places where water is a really limited resource, where it's not a good idea for plants to open up those stomata very much during the daytime because they're going to lose a whole lot of water. So we're talking about things that live in very arid and typically quite warm conditions, like, for example, a desert. Now, in this case, it's not going to be a great idea to have those stomata open at night because too much water is going to be lost, and that cuts down on the source of electrons. So what these plants do is they go through what is known as CAM photosynthesis, and that's short for crassulacean acid metabolism. There's no crassulacean acid made here. It just is the name of the genus of the types of plants that this was first discovered in. It was first discovered in the jade type of plants. Now, what these plants are going to do is they're going to close their stomata down during the day, so they're not losing too much water by transpiration. And what they're going to do is they're going to open their stomata at night, and they're going to do this carbon fixation phase at night. So they're going to be grabbing carbon dioxide molecules with PEP carboxylase, kind of like C4 photosynthesis, and they're going to be grabbing those carbon dioxide molecules and putting them in these C4 carbon trucks, and then taking those carbons and waiting till the sun comes up, and then they will be offloading those carbon dioxide molecules and delivering those carbon dioxide molecules to rubisco, and rubisco can then take that carbon dioxide molecule and bring it into the Calvin cycle, where we can make those carbohydrates during the daytime. So the carbon fixation phase happens at night, and then we wait to do the Calvin cycle during the day when we have the ATP and NADPH coming from the light reactions of the Z scheme when the light is pushing those electrons around. So in CAM photosynthesis, we're still doing carbon fixation. It's very much like C4. We're just doing the carbon fixation phase at night. We're pressing pause on photosynthesis, and then we're going to give that carbon dioxide to rubisco when the light reactions are able to run, and we can get the Calvin cycle going when the sun comes up and those stomata snap closed. So we see plants living in very arid, dry environments that do this, like cactuses, and up in the top there, pineapples are going to do this as well. It's an adaptation to reduce transpiration and prevent that massive water loss during the day. So those are the three main types of photosynthesis: C4, C3, and CAM.